Welcome to Pawpaw TV. Today we present an original program, One-on-One with Jerry McLaughlin. He discusses his life's work with Richard Lund. Today's program is brought to you by Gogees.com. Gogees.com. Your eyes will love you. This material is for educational purposes only. If you are sick, see a doctor. Maybe you can find one that speaks herbal medicine. And now, One-on-One with Jerry McLaughlin.
And in that work, then, what led you to the Pawpaw? Since we're here for Pawpaw TV.
Okay. Well, actually, very early on, when I was about 4 years old, my father gave me some pawpaw fruits, and I ate them. He called them the Indiana bananas, and said these were like bananas and you could eat them. And this was during World War II, and I liked bananas, but you couldn't get them because of the U-boats were sinking the banana boats, and so I ate those pawpaws, and I ate too many of them, as I recall, and I got sick and I threw up.
Mm-hmm.
And I never forgot that. So I knew there was something that's very powerful in pawpaws. And then later, in 1976, we were screening the pawpaw trees. The leaves and twigs that we'd obtained, I believe, originally from Alabama, and this is in a project with Drs. Chang and Dr. Cassidy. And we screened them, and they showed biological activity in mice, in the anti-leukemia screen in mice. Of course, we don't use that screen anymore, but at any rate, from that, then I was furthermore convinced that there was something in there. And finally, in about 1980 or so, I put a student on that project, and they started working on it. By 1982, we knew we had something that was very useful. Actually, at that point, I'd had Eli Lilly screening the extract, and actually they screened it against insects because it was showing some toxicity and biological activity against brine shrimp, which are a very interesting indicator, little sea monkey organism. And we found that the extracts were quite active at Lilly against 5 of 7 indicator pests that they had. And a fellow called me, and I can remember it was in October of 1982, and he said, "Jerry, what the hell is this stuff?" And I said, what do you mean? He says, "Well, we're killing mosquito larvae down at say 10 and even less parts per million with this extract."
Wow.
And so let's see what's in there. Well, it took us 3 years to isolate the first active compound. And one of my students, Kent Ruprecht, did that project, and by 1986 we published the structure of that compound and filed our first patent application.
What was the name of that compound?
And we named a simicin, named after a simina. The scientific name of pawpaw is a Simina triloba. So a simicin was our first compound, and we then proceeded to isolate with more workers, of course, a total of over 20 years of labor working on the plant extracts. We isolated over 50 of the compounds that are very similar in structure to a simicin. And it turns out these are highly unusual long-chain fatty acid derivatives that form tetrahydrofuran rings in their structures. All in all, we've isolated over 200 of those compounds from pawpaw and its cousin species in the Ananaceae family. And so they're now called the Ananaceous acetogenins. So those are the magic bullets, so to speak, that are in pawpaw. It's a mixture of over 50 complicated acetogenin structures, and we have proven every one of those structures down to their last little detail of stereochemistry.
And that takes a lot of work.
That's a lot of work, that's right. Over 100 publications in our scientific journals.
That period of time, then, the research was funded by whom?
We had money originally. The first grants was from the LaGrange County, Indiana Cancer Society. So I really encourage people to help support the American Cancer Society, because some of that money at least gets filtered down to the universities where it does some good and helps young professors like I was back in 1973 to get started and actually pursue a career to help with cancer. And then originally we had contracts from the National Cancer Institute for 6 years. I was with Dr. Chang, Dr. Cassidy. And then after that, I was able to get 13 years of funding from the National Cancer Institute's R01 program. That's the researcher-originated grant program. Those are the toughest grants to get. And you have to go through peer reviews, and very hard to get those in the first place, and then further, very difficult to get them renewed. But I was able to get 13 years of funding from that. And then we had other sources of funding as well. Purdue University, of course, helped, and we had, with Dr. Chang, a new drug discovery contract for a 5-year period. And so all the stuff all together, I think, totaled about $5 million.
Wow.
So this project is not cheap. So the results that we've come up with are the results of the taxpayers giving us money.
That's terrific.
Yeah.
Now, after you discovered these compounds and realized the stereochemistry, did you provide any measurements of how they acted in comparison to other compounds that were active against cancer?
Initially, one of the major contributions that my group made was that we came up with 2 very simple biological assays. One of them was killing brine shrimp, which are sea monkeys, and you can grow them in salt water, and they're very susceptible to pollutants in their environment. So the pawpaw compounds are toxic to the shrimp, and we kill them. The second one was the growth of tumors on pieces of potato. And there we took discs of potato tubers. Red potatoes happen to work the best. And you can inoculate them with Agrobacterium tumefaciens and grow little tumors on potato discs. We call that potato disc assay. So I worked with what we call shrimp and potatoes and came up with these compounds because anti-cancer compounds inhibit the growth of those tumors on the potatoes. And this allowed us to proceed without having to use animal studies. And also the cost is really cheap to do these biological assays. You're not having to maintain colonies of animals or grow plant or grow tissue cultures of animal cells, which in turn requires fetal calf serum and a bunch of very expensive ingredients. Comparatively, if I would have our extracts assayed at the Purdue Cell Culture Lab, for example, at that point it was costing about $120 to have one material assayed at the cell culture lab through 6 cells, 6 cell types. Whereas in my own lab I could run these assays for pennies. And this allowed me to use the money that I was given to do the chemistry instead of doing more, instead of paying for the biology. So I put the money into the chemistry, and I think that really paid off.